I had to use a handheld because I don't have a belt. So, show of hands, how many of you know somebody who's had cancer treatment and got sick from the treatment? You see, right now, we have to cut it out, burn it out, or poison it, and we have to harm good cells with bad cells. Novelogic is dedicated to developing cancer treatments that should not make patients sick to get better. It works something like this. Did you know that pregnancy and cancer actually have something in common? When a woman gets pregnant, there's a shed protein from the placenta that in essence puts her immune cells to sleep and allows the baby to grow. That's a great thing. However, did you know in cancer, it's the same shed protein that puts a patient's immune cells to sleep and allows the cancer to grow? That's a bad thing. Imagine if by design we could develop a drug that only binds this shed protein and allows a patient's immune cells to come back and fight cancer like it's supposed to. You don't have to imagine anymore. We'd like to present to you today the interceptor approach. You see, this shed protein is found in both blood cancers as well as solid tumors and some diseases, and of course pregnancy. So it is a very wide and large market opportunity, and unfortunately, with an aging baby boom population globally, a growing one. It's important to note that this is not found in healthy people. The one on the cell surface is found on healthy cells, but the shed one is not. The oncology market in immunotherapies is staggering. They assume that 60% of all cancer patients will be treated with immunotherapies. It is expected to grow to 124 billion per year in just 4 years, and that's a growth rate of 14.5%. We have differentiated IP. It has been internally developed. It did not spin out of an academic institution. Our sole inventor is here today with us. It has been 100% and assigned to the company and our shareholders, and we've filed in multiple countries, and we've also just extended into China as well. So how is your immune cells supposed to work when you get cancer? Basically, your NK, natural killer cells, and T cells have some receptors on them, and they typically dock to its matching protein and kill the cancer. However, as cancer advances, there's a protease scissors that actually cut off this protein, and it gloms up your receptors. They cannot dock. The second thing that causes immune evasion by the shed protein is it also signals further immune suppressor cells. So it's a doubling down on the immune system, and a cancer happily grows. So you can imagine with one drug, we will show you how the interceptor works. It gets injected into the bloodstream and binds up only this shed protein, and therefore we believe allows docking and killing and prevents further immune suppression and kills cancer. The third big important point to this is that we believe we avoid potential toxicity by design. You see, our competitors' approach are trying to stop this from coming off or binding on the cell surface and signaling the immune system. However, theirs does not differentiate between a healthy cell and a cancer cell. It is in your gut, it is in your endothelial cells, your thymus. So if you put an antibody that doesn't discriminate, it will bind both cancer and healthy cells. Probably work in a mouse. Going to humans is going to be tricky. We think there's a high potential for toxicity. As you can see, Novelogic's antibody only binds after it's shed and doesn't have to worry about discriminating between a healthy cell or a cancer cell. And we've avoided potential toxicity from the get-go. The competition, as you can see, Genentech, CanCure, Innate Pharma, Dana-Farber, all bind on the cell surface, and we are the ones that only bind the shed protein. We've got evidence that it works. We inhibited tumor growth by 53% in mice with only a fraction of the immune system in a solid tumor. That's prostate cancer. The interesting thing, how many of you know that you can now give mice a human immune system? So we did the mouse model where we actually gave it a human immune system. Now we could see, does it work with T cells and NK cells? And I'm happy to announce that this year we inhibited tumor growth with both NK and T cells, and we had hit efficacy with our monotherapy as well as in combinations, including one that with an immunotherapy drug that's already on the market, something that large pharma is actively seeking, both monotherapies and combination therapies. Because we have no overlapping mechanism of action, we think it should safely combine with some of these therapies and even some chemotherapies if a combo is necessary. The other thing to note is we saw no visible drug-related toxicity in any of our mice. So, how does a typical biotech company exit? It's milestone driven, so the more milestones you hit over your timelines is when a licensing deal or acquisition of your asset becomes interesting. We've already finished the discovery phase. We are now have a humanized antibody ready to go to the preclinical phase, manufacturing, toxicology studies, some further bioassays. So we're raising $7 million US. The important thing to note is we're from Canada. We get a substantial amount of non-diluted funding, including SHRED tax credits, NRC IRAP government funding, and we can also capitalize on that 25 cents on the dollar exchange rate as well when the work is done in Canada. So that will take us through to the preclinical work up to an FDA filing and approval, and that's where we think our next bump will be in valuation. And of course, our goal is to get to first in human. 18 months away, we could be first in human. At that point, we would do a raise and a clinical trial. Here's some comparables. How many of you have ever invested in immunotherapy? You've seen the returns on this. This is just some from 2017 in the discovery phase, which we're now out of. We've moved to preclinical. You can see that it's double digit in upfront millions, and then the deal totals in the $1 billion. Watch what happens after a phase 1 clinical trial. It can go up to multi-digit multi-million exits with $1 billion back end. The management team. I'm going to ask my team to stand. They're here with us today because we wouldn't be here without a team. I'm the CEO. Just became the CEO 2 weeks ago. And where's Dr. Cheney? Dr. Cheney is the sole inventor. It was his concept, his idea. He drew that diagram for me, and we got to work. So both of us have created the company and moved on. Dr. Mark Bazet just joined us. He's our immunologist. He is going to take the drug through to the clinic. Matthew Sebastiani, there we are. Come and talk to us at the table. Lots of questions on both the deal and the science. And also Dr. Neil Clendenin. Anybody know Dr. Neil Clendenin? He had done an internship up here at the University of Washington. He is an MD, PhD in pharmacology. He holds the record, I believe, for taking a drug from inception to the market in 36 months. So we've just had him join on as our Chief Medical Officer. Scott Cormack, from former CEO of Oncogenics, which was a prostate drug. He's been through a successful exit as well as IPO, and he's been a VC for the last 5 years. And we also round out our advisors both on the regulatory and clinical side as well. Traction to date, we have evidence with our humanized antibody, both monotherapy and combination. We've expanded the team, we're ready to go preclinical. We've got differentiated IP and filed globally. We just recently got issued our patent in Japan, and now Australia just a couple of weeks ago, and many more to come. We've been both voted Best Pre-Revenue Company and Most Innovative Company by the Vancouver Angel Group. They have supported us. I wanted to say a shout out to Angels. If they could rise to, they're part of our team, and say thank you as well if you're in the room. We had about 7 last night. Thank you so much. As I mentioned, wouldn't be here without you. So please visit our booth. I have some copies there as well if you didn't get one in your bag. And we're basically here to invite you today to be a cancer interceptor investor. We're a novel company. We have a novel approach. We're Novelogics.
Thank you so much. We've got time as we finish up here, just before our lunch break. So we do have a question right in the middle here. It's going to take a little bit for the mic. We'll go up to Paul here to get us started. We pass the mic to the middle here.
Fascinating presentation, and I think it's a much more modern take on how to treat cancer.
Thank you.
Okay, so with the shed proteins, how often do you have to take this drug? Because aren't proteins being produced all the time?
Where is Dr. Cheney? Can I get you to answer that quickly, or can you answer it at the booth better?
I can answer it at the booth better.
All right.
Okay.
I'm gonna refer that one to Dr. Cheney, okay, or Dr. Mark Bassett. Will it attack leukemia? It is found in both blood cancers and solid tumors. Dr. Cheney?
Yeah, it should work well in blood cancers and many solid tumors as well.
And the due diligence process is getting started and ongoing, and will be.
Yeah, so we've also got our
So-
DD team in place as well, so we are starting that process, and we're hoping to have the report at least in draft by September 30th. We need one more volunteer, if there is anybody in the room as well, just for reference checks.
And go ahead.
Yeah, so my question is basically, I think, one that was just asked, and that is, since the antibody only recognizes the soluble or the released, the cleaved form of the MIC-A, MIC-B protein, that suggests that in order to keep the patient, you might get regression of disease, they may even be disease-free for a period of time, but then at some point, if you don't keep giving the antibody, you're not really doing anything to drive the anti-tumor immune system. I mean, you get ADCC, but you don't get T-cell activation, right?
We're actually, I'm gonna get Dr. Cheney to talk to you about that at the booth, but we're not going to look at ADCC. Basically, what happens is that protein comes to the surface, and at a point it starts shedding, but you still have some on the surface. And if you can get rid of the immune evasion, we believe the docking is occurring and killing the cancer. And so it's this dynamic process of getting on top of it. And as long as you're removing what is causing immune evasion, you should get on top of it and put your cancer, be able to kill your cancer with your T and NK cells coming down and docking by themselves, without overstimulating them or causing autoimmunity. A lot of them are trying to trigger that immune system.
Other questions? Our time.